Commercial lipsticks were first crafted in France in 1869 using beeswax and animal fat. Today, applying lipstick is a universal daily routine. This diagram shows the biological composition of modern lipstick. Beneath the pigment, the bullet is packed with water, lipids, and proteins. These specific ingredients create a highly nutritious environment for microbes. Every time that waxy surface glides across a mouth, it picks up dead skin cells and whatever bacteria happen to be lingering on the surface of the skin. After just a few uses, a cosmetic designed strictly for beauty contains all the necessary components for a thriving microbial community. Microbiologists are actively investigating this environment to identify the exact ecosystems that develop on our makeup. Researchers Inyang Henry and Kathryn Bassey from the University of Cross River State in Nigeria decided to investigate this cosmetic blind spot. They published their findings in the Journal of Contemporary Academic Research and Methodologies. Their aim was methodical, to isolate and identify the specific strains of bacteria living on used lipsticks collected from female students. Understanding this research is a necessary step in identifying risks within our daily personal hygiene routines. Researchers began by swabbing the surfaces of 10 different used lipsticks collected from students on campus. In the lab, they dipped each swab into distilled water and let it stand before shaking the mixture to create a liquid stock solution. By repeatedly diluting the sample across a series of test tubes, scientists separated the bacteria to isolate individual colonies. They plated this diluted sample onto sterilized nutrient agar and incubated it for 24 hours to force bacterial growth. To identify the resulting pure isolates, researchers used a battery of biochemical tests, including gram staining and catalase reactions, to categorize the specific strains. These cultivation methods are the only way to make an invisible microbial presence visible and quantifiable. This bar chart shows the quantitative bacterial load discovered across the 10 samples. The counts ranged from 180,000 to a high of 650,000 colony-forming units per gram. The most prevalent contaminant was Staphylococcus, which appeared in nearly 28% of the samples. The study also identified Pseudomonas, the pink rod-shaped bacteria seen here, and Rothia species. The researchers found further contamination from Klebsiella, E. coli, and the long, twisting chains of Streptococcus. Because these organisms are associated with human skin and oral microbiota, the data suggests the lipstick acts as a direct vector for cross-contamination. 3 primary behaviors drive this bacterial buildup: direct skin contact, exposure to the environment, and sharing cosmetics between users. This contamination is compounded by the chemical limitations of the product itself. Over time, preservatives can degrade, making the waxy medium more susceptible to microbial growth. Manufacturers are responsible for implementing effective preservative systems and conducting regular testing to ensure the product remains stable throughout its shelf life. Preventing contamination requires a dual approach: advanced manufacturing chemistry combined with strict personal hygiene boundaries. The most actionable rule derived from these findings is clear: users should avoid sharing lipsticks to protect their personal health. Ignoring this risk has physical consequences. These bacteria don't just pose a health risk, they actively alter and deteriorate the chemical makeup of the cosmetic. By utilizing meticulous laboratory methodologies, Henry and Bassey illuminated a public health issue often overlooked by consumers. Their study demonstrates how microbes can thrive on the everyday tools we use to present ourselves to the world. True hygiene must extend beyond the body, applying equal scrutiny to the products we carry and apply every day.
Submit your manuscript today.